Gene regulation and mutation. Gene expression is regulated by the cell, and mutation can affect this expression. Gene regulation is the ability of an organism to control which genes are transcribed in response to the environment. Prokaryote gene regulation. In prokaryotes, an operand often controls the transcription of genes in response to the changes in the environment. An operand is a section of DNA that contains genes for the proteins needed for a specific metabolic pathway. A regulatory gene turns on and off transcription. Eukaryotic gene regulation. Eukaryotes have many more genes than prokaryotes. How is transcription controlled? Transcription factors ensure that a gene is used at the right time and that proteins are made in the right amount. The complex structure of eukaryotic DNA is also regulates transcription. Some transcription factors guide the binding of RNA polymerase to a promoter that determines which protein will be made. Hox genes. Hox genes code for transcription factors and control differentiation process through which all cells become specialized in shape and function. Hox genes are used in embryo development and are active in different zones of the embryo and control what body part will develop in different parts of the embryo. Hox genes are responsible for the general body pattern of the most animals. RNA interference. Another way eukaryotic genes are regulated is RNA interference. Small segments of RNA bind to mRNA and prevent several parts of mRNA from being translated. RNA interference can stop the mRNA from translating its message. Mutation. A mutation is a permanent change that occurs in a cell's DNA. A mutation in a gene sequence can change the protein that is made. A mutation in DNA changes the amino acid base sequence. A different amino acid or series of amino acids are incorporated into the protein that is made on the ribosome. The result can be malfunction of protein. Mutated proteins often do not work. Remember, the shape is very important to its function. A mutation can affect a single nucleotide or large segment of DNA. Type of mutation. Point mutations. Point mutation, a mutation in which one nucleotide base replaces another. Point mutation affect only one codon, so they affect only one amino acid in the peptide chain. It may or it may not series of effects. What? It may have or may not have serious effects on an organisms. It depends on where the mutation occurs and how it affects the protein for which it codes. It can be harmless. Muscular dystrophy is an example of a disease caused by point mutation. Muscular dystrophy is progressive muscle disorder characterized by weakening of many muscles in the body. Types of mutation. Insertion. Insertion is the addition of one or more bases into a nucleotide sequence. This can change many amino acids in the polypeptide chain, thus changing the protein. It causes a frame shift in mRNA bases, thus causing the wrong amino acid to be added to the polypeptide chain. Types of mutation: deletion. Deletion: one or more bases is removed from a nucleotide sequence. A different amino acid is put into the peptide chain, thus changing the protein. Cystic fibrosis: a disease characterized by abnormality mucus in the lungs, intestines, and pancreas, in an example of mutation caused by deletion. Frame shift: both insertion and deletion result in a frame shift. A frame shift is simply a shift to the left or right in the nitrogen bases resulting in a new codons. A frame shift changes the number of bases in a sequence. Chromosomal: point mutation, insertion, and base deletions affect only a single gene or segment of DNA. A chromosomal mutation affects the entire chromosome. Chromosomal mutation change the number of genes in a chromosome or the organization of the chromosome. We will cover more of this with genetics. Fragile X. An example of mutation is Fragile X. Fragile X syndrome is due to many extra repeated CGG units near the end of the X chromosome, making the lower tip of the X chromosome